Hello, welcome to today's cluster seminar. Our speaker today is Gloriana Chaverri, who's originally from Costa Rica and also did her bachelor's degree in Costa Rica before actually undergrad, sorry, also master's. Before she then moved to Boston to do her PhD on tent making bats. And then from there she did a postdoc at Michigan State University, then went back to Boston, and then at some point ended up in the Universidad de Costa Rica, where she has been ever since and does really, really amazing research, given actually relatively limited resources. And she'll tell you all about the really, really exciting and cool stuff she does. So looking forward to it. Great to have you here.
Thank you, Chini, for the introduction. So today I'll be telling you about some of the studies that we've done, looking at how individuals differ in personality and other traits I'll be telling you about, and how that may be translated to some of the collective decisions that they make. All right, so we often, when we think about animal groups, we often tend to think about them as if they're all the same. All individuals within a group are the same. But of course, I don't need to tell this sort of audience that we know that this is not the case. Individuals may differ in their size, their physiology, and also they may differ in their behavior. Some individuals are more exploratory, others are more sociable, and other different traits that, we already know that many animal groups have been quantified in this sense. So what do we know so far about how these individual differences within these groups translate into collective decisions? So I'll just give you a couple of examples that I found really interesting that may be really relevant to the studies that I'll be telling you about. In the first case, there's been several studies done on homing pigeons that have tried to quantify how individual differences may translate to collective decisions. And in this case, this species has been studied in terms of how do individuals fly when they're by themselves, and how that translates to whether the group follows certain individuals or others. So in this case, studies have shown that some individuals fly very quickly, but when they're by themselves, whereas others are much slower. So when they try to quantify the decisions of the flock, they know that certain individuals would prefer certain directions of travel. So these are the slower ones, this one is the quicker one. And studies have shown that the flock tends to follow the quickest individual. In Chacma baboons, we also, in the context of where this species wants to go forage, let's say we have different foraging patches, and we know that Chacma baboons form these hierarchical societies where I'm very simply putting this like the dominant individual compared to the subordinates, and how they make decisions depending on where they prefer to forage. So let's say the dominant individual would prefer to go to this patch, the subordinates would go to this other one, but when they make collective decisions, they usually go where the dominant will prefer to go, even though this comes at a cost to the subordinates. So this already shows very clearly, at least in these two examples, that we can see how individual differences and preferences may be driving collective decisions. So we do have examples about this, and I'm not showing a lot of other studies that may be also showing very clear evidence of this phenomenon. But I think quantifying also individual differences is really complicated because we have such a large suite of potential differences in let's say, personality or different traits of individuals. And which one of these really translates into, how groups make collective decisions? Which ones of these are really relevant for making these decisions? And also the way we quantify some of them is really complicated. In some species, it's difficult to come up with really biological or ecologically relevant tests that we can conduct to really see the differences between individuals. And then also when we try to quantify collective decisions, I think this is, for many species, is something that's very complicated to do. For example, the species that I studied that I'll be telling you about, it's so small, so you cannot really follow them very accurately if you put tags, because they're just too heavy for them. We do have made a lot of progress in this sense with these tags that allow us to measure their speed and the position of individuals. And I think a really cool example is also done on homing pigeons. So you can track them quite accurately and see how they making decisions when they're trying to avoid predators, for example. But then, so the study species I've been focusing most of my research on is the Spix's disc-winged bat. It's a very small bat, only around 4 grams. And the name comes from these really cool structures that they have on their feet and hands. And they use these structures, we've seen in detail here, to attach to the smooth inner surfaces of the plants that they use for roosting. So this is a species that lives in relatively small groups around, let's say, a median of 5. They also have all-sex phyllopatery, so pups, when they're born, they stay within their natal groups. And so this results in very high relatedness among individuals within the group. And groups are also very stable. Individuals stay together for several years without changing group composition. A lot of the studies that I've done have also tried to focus on understanding how this particular resource, this is the typical tube where the bats would go into, and these are when the leaves are developing in plants like heliconias, calatheas, and bananas, they have this tubular shape. And the thing that these tubes remain within this preferred range of size, around 7 centimeters diameter of opening, for even less than 1 day. So the bats have to very quickly search for a new roost site. So what happens often is that the leaf opens up during the day, and at that time, then they become really conspicuous to predators. And so they have to quickly leave that roost where they're in, and very also quickly find a new suitable place to remain for the rest of the day. They often do this during the day, and during the day, bats are really, really susceptible to diurnal predators. So then they have to do this very quickly, otherwise they not only are subject to predation, but also, of course, they're wasting energy at a time when they're supposed to be sleeping or resting. So what we've done in several studies is to try to understand also whether they communicate when they're doing this process of searching for a new roost site. And what we've seen, this is a study done several years ago, then we see that the bats, when they're flying, in addition to echolocation calls, they're also emitting a social or contact call that we've called an inquiry call. They do that, they emit that call very often, and when a group member has already found a leaf to go into, then it hears one of these inquiry calls and they start producing the response calls. So once they do, the bats that are flying will go quickly into the tubular leaf that they're using at that particular moment. So in this video, I'll show the process because this is also very relevant for the remainder of my talk. And this is a camera pointing downwards, looking at the entrance of this plastic structure that they readily use for roosting. And then you'll see the bats flying around, and they have different colors because we put powder that allows us to recognize each one of the individuals. They're quickly flying. Then you'll hear one of the inquiry calls, and I'm going to show on this side also the sonogram, the spectrogram that shows that call being emitted at that time. And then when the bat goes in, you see the response call. So let's see the process. Yeah, so this is a very, very quick coordination that they do to enter this new leaf, a structure that they didn't even know that there was there in a flight cage where we normally do our experiments. So this is more or less the process that we're interested in trying to understand. The bats are within a leaf, something happens, maybe a predator comes or the leaf opens up, and they have to quickly start searching for a new roost while they're emitting inquiry calls, and then they select the roost that they are going into. So basically, this is the entire process where we're trying to see how these individual differences translate into collective decisions for the disc-winged bat. So which collective decisions I'm trying to describe for you today here, and especially in the context of these individual differences. So the first one is in the emission of inquiry calls as the bats are flying around as a group. The second one is in the process of exchanging signals, so the inquiry and the response call. That's the other collective task I'm going to be telling you about. And the last one is when the bats are flying as a group and they end up selecting one leaf over another. When I talk about individual differences, I'm going to focus on three very basic ones that we have addressed so far. The first one is in the differences in the contact calling rates that they emit. The first one is in the inquiry calls. So we know that certain individuals are more vocal than others, and this is very consistent. And then we're also looking at the differences between individuals. Some of them emit a lot of response calls when they find a roost. Others don't do that as often, or never, according to what we've recorded so far. And the other individual differences, this is a very recent study that we're doing right now, is in the process of roost finding, how different individuals may differ in how quickly or how exploratory they are when they're searching for a new roost site. This is the stuff that I'll present at the end of my talk. So just for context, I wanted to show you one of our field sites, the studies that we normally do in that site, and then also to try to understand certain aspects of the biology of this particular species that may be relevant for understanding certain phenomena that we've been studying. So our study site is in Costa Rica, right there, which is called Hacienda Barú. And in this site, we usually track all individuals and come like once a month to this site to try to sample them as often as possible during that month. And then in this site, we try to capture all the individuals every single month and try to fit them with transponders. And usually we have around 75 individuals within this entire 6-hectare plot. And then when we started doing our research, we were able to find 11 groups, but this year the number of groups increased to 15. So every time, so we go searching for the bats, if it's a new group, or maybe once a month if we need to measure them for different goals. Then when we find one of these tubes here, and if we know that there's bats in there, then we'll try to capture them. It's a really, really easy process, very easy to find all individuals within the roost, then place them all together in the cloth bag, and we just bring them or do whatever we need to do with them in the field. So once we've tagged them all and we just return to the field, we don't need to recapture them every single time. So what we do is use one of these extendable mirrors, search for the bats in there, and if they're within the leaf, then we just read them constantly to see which individuals are within the leaf. And it's very accurately tells us which individuals are within that tubular leaf. So we've done this already for 5 or 6 years, and the data that I'll present to you here is based on data from 2021. Basically, what I'm showing you here is this 6-hectare plot, and I'm showing the position of different groups within this 6-hectare plot. So each one of these little X's you see here represents a tubular leaf that the bats may be using as a roosting site. The dots, the different color dots, represent a group of bats, individuals that are sharing the same roost at the same time. And then the shaded area represents the territory, or the roosting range of that group, which is based on the 50% kernel density estimator. So I think 2 important messages are relevant from this figure. The first one is that the roosting ranges are fairly small, and the second one that there's very little range overlap. So to us, this strongly suggests that this species is highly territorial. And so this is one way in which they're also creating this, or reducing opportunities to be interacting with with other groups. So this is relevant for trying to understand the biology of this species. And then in this other graph, what I'm showing now is the social networks in the same positions where we already saw these other groups. So each one of these groups is shown in the social network, also based on data from 2021 for all individuals that were captured on 10 or more occasions. And so each one of these basically is represented. I don't know, I mean, I'm pretty sure everyone here is familiar with social network, the way we present them. But each one of these nodes here represents an individual. The line here, especially the width of the line, represents how often two individuals were found roosting together. So again, in this slide, what is really relevant is to try to show that the social networks are very close. There's very, very few exchanges between individuals, even after we've tracked them for an entire year and went searching for them for more than 30 days that year. They were never roosting together with individuals from other groups. And when they do so, like in this case, for example, the individual just permanently moves to a new group. They never go back to their original group. So this is also something interesting about this species. Okay, so let's go back to the topic at hand, and that's how these individual differences in the calling rates are maybe driving collective decisions. So the first important topic here to cover is that, do we really see differences in how many inquiry calls or response calls individuals are producing? So we've already done a couple of studies, published a couple of papers showing that there are, in fact, consistent individual differences in the amount of inquiry calls and response calls that they produce. So the first graph up here represents each one of these panels is showing one group. And each line here is an individual. So we see that, for example, for inquiry calls, there's a lot of variation within each one of these groups. But usually we find that individuals produce at least one inquiry call when we carry out these experiments or we release them in a flight cage. For response calls, it's a little bit, it varies a little bit more than this. So we see that in certain groups, no individuals are emitting calls, and there's one that emits those calls. Okay, and we see this very often in many of the groups that we constantly capture. And this, you'll see why this is relevant later on. Okay, so we find consistent differences between individuals in their calling rates. So now we're trying to look at how are these calling rates driving some of these collective decisions. And this is a study that was funded by the Costa Rican government. And it was done in collaboration with Damien Farin and Holger Görlitz when Max Planck Institute for Ornithology was still existing. So that's why I'm showing the logo here. But done with my postdoc Marcelo Araya Salas and my wonderful students, Nasa, Silvia, and Mariela. So there's a lot of light, but there's bats flying in here. This is a flight cage that we use for these experiments. And we have microphones set on the floor, and we're recording individuals constantly. So for the first step that we did in this study is to record the inquiry calls when the bats were flying by themselves. The second step is to record how the groups are behaving. So we released individuals that belong to their natural group. So this is the typical group we usually see. And this is represented by these illustrations that are the same color. And then we also created artificial groups. So we released individuals that belong to different groups, allowed them to fly and see how they were behaving. And after we did this later step, and based on the recordings from individuals when they were flying by themselves, we were also identifying calling events when we had these group flights. So what did we see? So how did the calling rates for inquiry calls differ between each one of these different treatments? So here I'm just showing how single individuals are behaving when they're by themselves, when they're in their natural groups, and when they are in their artificial groups. And this shows calling rates per individual. So what we see is that when bats are by themselves, they produce a lot more calls. They're constantly calling, whereas each individual, when they're found within their natural group or also their artificial group, call rates decrease quite significantly. So now then we say call rate per each one of these individuals decreases during these group flights, okay? So what happens for the entire group? So when we sum all of the inquiry calls that they produced, now all together, so when this individual is by itself, when the entire group is calling, when it's the natural group or when it's artificial groups. How are these in total? How many calls can we find? And now we see that when we compare the solo flights to the natural groups, they're basically emitting almost the same number of calls than when they did, like total number of calls when they did by themselves and now when they're in a group. But we see that for the entire group, when they're artificial groups, the call rate decreases a little bit. Okay. So now what we see is the call rate per group is the same for solo and natural groups. And of course, this means that individuals are changing their individual calling rates when they're together in a group. So we see then that bats are modifying their calling rates during these collective flights. So this is what they do when they're by themselves. Obviously, this is changing when they're all together in a group. Okay, so this second part, how bats differ in their response calling rates and how these may be driving collective decisions. And these are studies that I've done primarily with my colleague Maria Sagot. So the most basic question for us here was to try to determine if when we do trials in a flight cage and we have, let's say we have a leaf, we put a bat in there, and having a non-vocal bat increases the time that individuals need for finding a new roost. So we know that response calls are very important for other individuals to find this roost. So what we want to, in controlled trials, to see really if the time differs between how long it takes individuals to find the roost. So what we did then very easily just placed a leaf and a bat that was non-vocal or a vocal bat, release the rest of the group and see how quickly it took them to find the leaf. And of course, as we expected, when we had a vocal individual within the leaf, the group was able to find that roost much faster. So of course, now we can say very confidently that response calling leads to efficient roost finding. But then a very important question was arising. So what happens if we have more than one vocal bat? What would happen then for group coordination? Let's say we have two individuals in two separate leaves, they're both calling. What happens to the group? So for this, we did an experiment, again in a flight cage, where we had a speaker within one of these tubular structures, and where we were broadcasting response calls. Or maybe two leaves broadcasting response calls, 3 or 4. So we wanted to see what happened to the group. Would the entire group go to a single leaf, or would they alternatively separate in different leaves depending on their immediate selection at that moment? In this graph, I'm going to be showing you the number of roosts that are broadcasting calls, either 1, 2, 3, or 4, and what's the probability of group fragmentation based on these many roost broadcasting calls. And what we see is, as we expected, that when we increase the number of roosts where we are broadcasting calls, then it's a lot more probable that the group will split into different roosts. So I remind you about these differences that I pointed out before, especially for response calling, that many groups have individuals where we have never been able to record response calls from, and that's very common in many of the groups. Even like some may be relatively quiet, and one is like extremely vocal. So we wanted to try to test whether these differences in calling rates for response calls were somehow allowing groups to remain more cohesive if there are different options of where to roost. So for this, we only did the experiment where we had 2 roosts from where we were broadcasting response calls. So one of the roosts had low calling rates and the other one had very high calling rates. And so when we did this trial for the first experiment that I showed you, what we saw is that when this was the case, when the 2 roosts were broadcasting calls at the same exact rate, shown by similar videos here, what we found is that very often the group separated. So in 67% of the trials, the group would separate if calling rates were the same. But in this case, now we increase the calling rates in this other experiment, and we see that very usually the bats will go into that single leaf. So we see that the majority of bats, majority of individuals, when we have different colony rates, high or low, they will normally go to the leaf that has higher colony rates. And only 26% of the group split, and this is compared to 67. So I think this is really interesting, how these differences in the response colony rates within groups may be allowing the groups to coordinate a lot better if they find in these scenarios when they're both two or more individuals found different roosts, and maybe that will allow the group to coordinate and go into the roosts where higher colony rates are being emitted. Okay, so for this last part, where I'm focusing on these individual differences in terms of how bats are finding new roost sites and how this may be driving collective decisions. So I'm looking, this is a study that's very recent and we don't have all the data yet, but I would love some input if you have any. And this is why I'm also explaining methods a little bit more in detail for this study. So this is a project that's been funded by the Smithsonian Tropical Research Institute, and I'm doing in collaboration with Rachel Page, Adriana Maldonado Chaparro, Inga Gaipel, and also Silvia and Mariela, wonderful students that are doing all the hard work in the field. Okay, so, and this is a study that we're doing in Panama and in Costa Rica. So in the field sites that we have in Costa Rica, we usually find very high availability of roosting resources, whereas in Panama, they're really, really scarce. So this also creates this very interesting scenario in which maybe groups are making different decisions based on what they usually find in terms of roost availability. And when we see photos of the two sites, I think it's very clear, like there's so many potential places where the bats could be roosting, and they're all really, really aggregated, whereas in Panama we find one, lost tubular leaf there, but they're very, very scarce. So here what we're trying to look at is whether individuals are different in some of their cognitive abilities. This is something for which we don't have the data yet, but we're looking also at differences in the personalities as they're searching for new roost sites. And then whether these differences may be driving group decisions, especially where they decide to roost. And the differences we're looking at is the latency to enter a new roost site. So we make one leaf available within a flight cage and many others unavailable, and see how quickly the individuals are able to find this leaf that's available. We also see at how exploratory individuals are. So how many leaves are they checking as they're trying to find the one that is available. And the last one is spatial memory. And for this one, we still don't have the data, but also I'll show you what we have so far for these. So here first we want to see which is the individual that first finds the leaf, okay? So that's one of the first data that we collect. So we see which is let's say the leader. And we want to see if that leader is also the one that takes less time finding that suitable leaf when it's doing the individual trials, or also during those individual trials, whether it's the bat that's more exploratory. But for exploration, we're measuring different things. So we want to see how many unique checks they do on leaves, okay? So if they check each leaf just once, of course, that's a number instead of having to go check always the same leaf. That's another measure we're trying to do. Then how many checks overall they're doing on the potential roost sites that they may be using, and how often they repeat the same leaf, even though maybe often it's not available. So we call this how stubborn the bats are. But we are also controlling these for how long it took them to find the leaf. So if they check a lot of leaves and it took them really short times to find the suitable leaf, then of course these values will be higher just because it took them less time. So we're also controlling for those issues. So we try to also summarize, and I'll explain this a little bit better afterwards. We also try to summarize the values for each individual For latency, exploration, for these 3 measures, try to summarize it for the 5 trials where we do the learning experiments for the bats, and get 1 value per individual. And this is the flight cage that we use. So we have, these are the tubes that we use to test them. They're plastic again, and we know that the bats readily use them. And so in this video, this is the only one that I have so far, but you see the bats checking this leaf here, and then we have the color of the bat, so the yellow is the one that entered the roost first, and then the others followed. So again, the first thing we did for these trials was to let them fly as a group. We did it twice to see how consistently the same individual was going in first into the leaf or the tube. And then after this, we did the individual trials, and for each bat, we did 5 trials. We didn't do more than 5 because bats get really tired after flying for 5 minutes. So I think it's fair for the bats not to repeat that very often. And so far, I'm only going to show you data that we have for Panama. We haven't finished our collection in Costa Rica yet. In these graphs, what I'm showing you is each one of the learning trials, and for each of the values or the personality traits that we were trying to measure. So in this case, each one of these panels represents a group, and the different colors here represent individuals, or the different line represents individuals, and the ones in bluish represent the leaders, and the other the follower. So it was very consistently always the same individual that entered first in the first group trial, also did that in the second group trial. And this just shows how quickly they were finding the leaf that was available to them. Okay, so this is for latency. Then we also have this for exploration, for unique checks, how many of all the leaves that we had available each bat was checking. We also have that for total checks, and then also for repeat checks. And so with the data for each one of these measures, we try to summarize those in 4 different principal components to try to understand whether followers and leaders had different values for each one of these principal components, to try to summarize what happened throughout the 5 trials. And so we run with this a linear mixed model where we had our PC score for each one of these values that I showed you, depending on the order, the leader or the follower, and the random variable was the group. And we saw, in general, what we saw, even though the trends are not overall significant, and these are the model coefficients, what I'm showing you here. So if you remember model coefficients, if you have the variation overlapping here to 0, it means that the model is not seeing a significant effect. But at least, I mean, with the data we have so far, we have some trends, and I thought it was interesting to try to present some of those to you today. So what we see first is the number of unique checks and total checks seem to be higher for the leader. So this is showing the leader effect. So whatever is up here, like the higher values in this case, is showing that the leaders seem to be a lot more exploratory than the followers. And also, we see that latency values are lower for the leaders. So of course, leaders seem to be finding the roost much faster, and when they're doing the trials by themselves compared to the followers. And repeat checks, how stubborn they are looking at the same roost, we see that the leaders are not that stubborn. It seems like followers are a bit more than the leaders. So we can, at least with the data that we have so far, we can see that the leaders seem to be checking more leaves, and they also seem to enter faster when they're doing their individual tries, and then that translates into them guiding the rest of the group into the available leaf that they use for roosting. So the idea is to try to have this comparison between the populations that we have in Costa Rica and Panama, and try to see if groups depend on certain type of personalities more according to the availability of roosting resources that they have. So let's see what happens after we collect all of our data. Okay, so I've tried to show you some of these collective tasks that we've been able to record for many years in this study species, the disc-winged bat. and how these individual differences may be affecting or maybe guiding some of the collective decisions that the groups are making. And I find this topic of individual differences very interesting, and I think it goes a little bit like understanding which individuals are very vital for the groups goes beyond understanding or beyond this scientific curiosity that we usually have. Because I think like when we know that groups may be losing individuals, specific individuals often, even because they're more subject to predation, they end up in someone's soup or they end up in a museum collection. And so what we also would like to understand eventually is how the loss of these very vital individuals for the group may be affecting the rest of the group that very strongly relies on these vital roles of these individuals. And especially now that we know that there are multiple other stressors affecting populations, I think this becomes a very relevant topic to try to understand further. And for this, after this, I like to help a lot of people, and not all of them are included here in this slide. But I would really like to thank all the people at the Max Planck that have made me feel like I was at home. The Zukunftskolleg for giving me a home while I was here. Like, they funded my stay here, and they were also a wonderful family. But very especially to Chini for making this all possible and being such a wonderful hostess. And thank you very much. Questions?
Thanks for the great talk. I guess I have the obvious question, which is the leader also the one that is doing the response calls to attract the other bats inside?
Yes, very often we find that, and it's for this study that we're doing right now with the Smithsonian. We haven't been recording the calls, and my students are very, very frustrated that we haven't done so. But we often find, like in other studies, we've shown that the bats that are a lot more vocal in terms of response call production, they're also the ones that are more exploratory, and we've seen that with putting readers on potential leaves that they may use for roosting. So yeah, I think there's an association, but yeah.
Cool. So you think they have, so I'm a social insect person, so I can't help but thinking about division of labor. And so you think these bats are actually specialized for this task?
Yes, this is something that we always wanted to try to answer. And I think there's also interesting to see, if we get the loss of one of these more vocal, more exploratory individuals, what happens to the group, whether another individual takes over. And I think that's a very interesting question we haven't been able to address as well. So.
I was curious how individually unique the calls are, the response calls or the contact calls. And do you think that has any influence on their preference?
Yes, so they are individually, so they have signatures, very clear signatures for both types of calls. And we've done experiments to see if they're able to recognize each other in different tasks. So the first one is when they're flying, whether they can recognize inquiry calls from their group members, and they seem to do so. So they prefer to join the side of the flight cage where we're emitting calls from their group members. Also, when they're flying and they have to select a roost, they would go primarily into a roost that's broadcasting calls from a group member, the response calls. But when they're inside the roost, they don't seem to be able to recognize, or at least they don't emit response calls more to group members. So, we also published another paper where we thought, maybe the acoustics of the leaf is, somehow affecting how they recognize each other. And, the leaf really is affecting a lot the signals that the bats receive. But we don't know if this is the cause of them not vocalizing more to group members. So we're still looking into those differences.
Sorry, just to follow up, what types of calls are you broadcasting in your experiments then?
Both the inquiry and the response calls.
But from which individual? Like from individuals within the group?
Yeah. From group members and non-group members.
Okay.
Yes.
Yeah, I didn't totally understand the full setup.
Okay. Uh-huh.
So when you do the experiment, you have all the potential roosts. It's a mixture of or each speaker is playing multiple types?
For the experiment that I showed you. Yeah. So no, that experiment. So because we were not able to get response calls from all the individuals within a group, it was very risky to try to do that. So what we did is, because we know they go into a leaf, even if it's a response call from a non-group member. So even though they do not prefer that, they would definitely go in there. So we were broadcasting response calls from non-group members in that experiment. Yeah.
Thank you very much for the talk. It was really interesting. I guess my question is a follow-up from the first one. Do you know if they have some dominance hierarchy for, I don't know, food or mate or something, or related to age or something that might be related to this personality / inquiry call, like more experienced individual or dominant individual have the choice of which leaf they can want to choose or something, like the baboons experiment you
Yes.
about.
We don't have an idea yet if they have some sort of dominance hierarchy. This is something that we tried to address, but it was like very indirectly in how they position themselves within the tubular leaf, because we know that they consistently choose the same position. So the one that's on top is usually the one on top, the ones in the middle, usually the ones in the middle, and the bottom, again, very consistently. So we're trying to understand that, but of course we don't have like cost and benefits of each position to try to see, okay, so the center should be the preferred one, so the dominants are there. So we don't know that yet. But also, like whether they would be able to attract or force the group to go into the roost that they have already selected, we also don't know that. So no idea if we would be able to find something, but that's definitely interesting.
Thanks.
Thanks a lot. That was super cool talk. I had two questions for you. I wanted to ask, and I think you already talked a little bit about this in response to Edward's question, but did you find any audience effects for the recruitment call, the response call, where you had this experiment where you had unfamiliar individuals together? Because they're territorial, it would be surprising to me if they would produce those response calls if they're only around unfamiliar conspecifics.
So we're testing audience effects right now, but not exactly in that context. So that would be interesting because we were placing like familiar individuals within the roost. So let's see, because this is one of my master's student projects. So for example, if you want to see whether they respond more when they're together with another bat and when they're hearing inquiry calls, and they always hear inquiry calls from their group members. So either they're by themselves or they're together with another group member. But we've never tried that with completely unfamiliar or external groups. Yeah, that's interesting.
Might be cool to see.
Yeah.
And then my second question was, you said so they rarely change groups, but when they do change groups, why do you think they change groups? Does anyone know what happens?
That's such a fascinating question. There's most often we see that they change groups when the group just gets very large. And so let's say we have groups, they're usually, let's say 5 or 6 individuals, and when they're all born, let's say March, we have all these new pups, and the groups are like, oh, now 11 individuals, and then usually one of the females with the young of the year leaves that group. So, for me, it's really fascinating that they do it together, like body dispersal, more or less. So I think this is really fascinating. So this is most often what we see that, or when the site has lost its quality very significantly, like a tree fell and they lost on the majority of the leaves, some of the group members will remain, whereas others just say, no, this is it, we're leaving. So,
cool. Thanks a lot.
Yeah.
Thank you for the talk. Being a communication engineer, I'm really interested in the communication side of it, especially redundancy as a characteristic. So we can do it qualitatively. That means you can improve your communication message, how you send it. You can do it quantitatively. Have you in your experiments, when you're trying to replicate this through artificially producing these calls, made them repetitive in some way? If you found a group where median of repetitiveness was 2 or 3, and then you just peak that number to 5, do you see any improvements there? What are your thoughts?
In the number of calls
Yeah.
that they, no, we never tried it, something intermediate. Yeah. Like, that's an interesting point. Yeah, we've only done it like quantitatively in the sense of, we know that this individual emits like 1 or 2, and this is what we broadcast. Yeah, is this your question more or less? Or wait? My biologist brain is like, ooh.
Thanks for a great talk and amazing system. I really should read more about it. I have a question about this similar call rates when flying by themselves or in the group. Do you try to see if this call rate is somehow facilitating the response calls in a better way, or is it not related?
I think, so the calling rates when they're flying, I think it's primarily helps them stay together as a group, and this is something that I really would like to look more into, because why are they like emitting a given number of calls as they're flying together? I think it may be because the speed that they're flying at, or that they somehow they need to make sure that a certain calls are being produced as often as possible so that they can remain together. But we haven't looked at whether that causes differences in terms of the response calls that are emitted.
From the leaf.
From the leaf, exactly.
Okay.
Yeah.
Cool.
Yeah.
The areas seem really small, right? 6 hectares or something, you said? And
yeah,
I'm just wondering about the foraging, and they must be meeting each other all the time during foraging, and is it way bigger than the distances between the roosts, or how to imagine it?
So I've never done anything on foraging. There's a colleague that tried to do that, and they, definitely the foraging area is much bigger than the roosting area. And we think the groups overlap also, but we really don't have a lot, of information, especially because of the problem of tracking such a small species. So, on foraging is still looking at that. Yeah.
Thanks a lot. This is a bit linked to the previous question. I did not fully understand the social organization. So they are territorial. They can fly on their own. They can fly with other group members, like with members of other groups. And or was it due to your experiments? Like you were taking individuals from different groups, put them together, and this is not something that would happen in real life.
Yeah, so we created this more artificial situation in which they were flying with other group members. But when they're coordinating to search for a new roost site, I think they only do it within their own group, basically because of they can recognize each other and they prefer to fly with group members. But in the flight cage we put them with individuals from other groups and trying to see how they coordinate and achieve all of these tasks. But and it was interesting in a way because we saw that there were a lot of chases, not all the time, but once in a while we saw that when we were using these mixed groups from different groups from other areas, that they will sometimes fight with each other. So I think this is not something that they do very often.
Because it kind of follows up on this a bit. It was about this intergroup conflict in that these territories are so stable. Do you have any plans for looking at this, and could there be a role for these followers and kind of more maintaining the territory while the other is finding new roosts? Have you guys thought about those kinds of things at all?
Mm-hmm. Always the question is how we would do that without. I mean, I think every year there's a possibility of finding smaller and smaller tags. So maybe soon we'll be able to answer some of these questions.
What would be the point to be territorial? I don't see the point. I mean, if you said the leaves are everywhere, right? So it's just this group belongs together, and so it's not actually territory, but it's just which group should be together and not be intruded by others.
Yeah, so there's, access to resources, definitely, especially because in some areas, there are a lot of tubular leaves that they could use, but, they could still face the option of not finding any in one given day. And then whether that really helps them stay in a cohesive group, I think that's also a very interesting question, whether this is another way in which they can remain with the same set of conspecifics. Yeah, we don't know exactly why. Yeah. Oh, sorry.
More questions, obviously. I was curious about the distribution of the inquiry calls. So, like, the call rate is per minute, I assume, but like, do you have much variability in that? Like, are they clustered or are they very evenly spaced calls?
They're, I would say, evenly spaced. Of course, we see some variation, but I would say that it's primarily evenly spaced. Yeah. So this is part of what we're looking at for this paper that we haven't published, like whether the gaps between calls are also very consistent, and we also don't see any differences even between like individual flights to group natural or artificial flights. So.
Any more?
I mean, when you put the artificial groups together, is there any aggression? Do you see any like difficulties of forming these groups?
Very rarely we saw chases. Like they were flying at a certain speed, and all of a sudden we see one, and then the other one is flying much faster. So we did get to see that, but I mean, we thought it was going to be more often, but it wasn't. So
not in the roost itself. There's not any. In
the roost, we don't see like they're super aggressive, but they emit these aggressive calls, and so often also an external group member will go into the leaf that's already occupied, and the ones inside like may leave the leaf, or the one that enters like wow goes out again. So. Yeah, I think there is some aggression, although this bat is very nice compared to other species that are a lot more nasty.
When it starts raining, it doesn't just rain inside.
Chini wanted to answer that question. This is something so exciting, but we have no idea what happens. And so for the majority of the tubes that they use, they don't accumulate a lot of water. Like some of the banana tubes, they really accumulate, so they have to leave. But in these calatheas or heliconias, they usually don't accumulate water. But how they're doing, because we've seen the bats when it's raining pretty badly, and I know the only time that I've seen a group do something like very interesting, it was the one on top changed the position in a way that it was covering the rest of the group. I'm like, but it's getting completely wet. So we don't know yet what's going on.
When you have those groups that are generated by having playbacks in multiple tubes, do they at some point realize, okay, we're basically split, we need to kind of rejoin and find the others?
Yes, this is something really interesting, but we didn't continue the experiment in a way that will allow them to rejoin. So we just, okay, they selected different tubes, okay, let's do the other experiment. But that would be really fascinating to say, oh, okay. And I'm not like the dominant maybe, and so I need to go find the dominant individual. So yeah, that's also something we haven't looked at.
Do you see like minimum group sizes? I saw in your network there were 2 individuals in certain groups. Like
yes. Yeah.
What do you think they need to be, or maybe they find roosts faster, or is there any advantage of being in sort of this median group size? Obviously, when it gets too big, there's no space, but
like the idea of
costs of being in a small group.
I think there are costs, like again, that we haven't measured yet. So maybe they don't have this extended network of ears or eyes looking for new roost sites, and then we also tend to see that those smaller groups use a much smaller roosting area. And then we also had, for example, for group 11, there was only the mom and a male that had been born several years ago, and they were always together, and then we saw the female die, and we're like, oh no, what's going to happen to that individual? So it merged into another group that was nearby. So they need to be with other individuals, but I don't know exactly whether having a lot more, which could also pose a coordination problem, right? As having all these different individuals to try to coordinate where you're going to be roosting. But yeah,
So given that you have the same positions of the bats in the leaf, and then you find that you have specific individuals that are more likely to enter first, I assume they don't really change position when they're in the leaf, so they would always end up in the bottom, no? And then you would expect kind of, right, that they're the bottom bat in the leaf.
Yes, although that's not what we found. So the most vocal individual is not the one at the bottom, so we assume that once they enter, they switch positions. But yes, we're not entirely sure.
Okay, in that case, I would like to thank Gloriana again for this meeting.
Thank you.